Chimeric antigen receptor T cell therapy, which involves the engineering of a patient's own T cells to eliminate tumour cells, has revolutionised cancer treatment. This highly personalised immunotherapy typically starts by collecting T cells from the patient's peripheral blood. These T cells are then genetically modified, often using viral vectors, to express CARs on their surface, before being expanded into millions in the lab. After quality control testing, the final step is the reinfusion of the CAR T cells back into the patient to attack cancer cells. CARs are modular synthetic molecules comprising 4 components: an extracellular antigen binding domain that is most commonly a single-chain variable fragment derived from a specific monoclonal antibody, a hinge region, a transmembrane portion, and an intracellular signaling domain that triggers cell activation. CARs redirect T cell cytotoxicity toward tumour cells expressing a specific antigen. When the extracellular domain binds to the target antigen on tumour cells, the intracellular domain mediates T cell activation and anti-tumour responses. Despite their considerable success, CAR T therapies still have limitations, including limited efficacy against solid tumours, CAR T cell-associated cytokine release syndrome, or CRS, and neurotoxicity, on-target off-tumour effects, and antigen escape. Additionally, using autologous T cells as a source material to prevent graft versus host disease, or GVHD, requires individualized collection, modification, and expansion processes that limit the scalability and accessibility of autologous CAR T therapy. Given these limitations, there is growing interest in exploring CAR natural killer cells as an alternative to CAR T cells due to their promising preclinical and clinical results. CAR NK cells offer unique advantages. They can be generated from various sources, as allogeneic CAR NK cells don't cause GVHD, making them attractive candidates for universal off-the-shelf immunotherapy. CAR NK cell therapies have demonstrated superior safety profiles with low risk for CRS and neurotoxicity. Furthermore, CAR NK cells identify and eliminate cancer cells through both CAR-dependent and CAR-independent mechanisms, boosting anti-tumour efficacy. Nevertheless, there are still hurdles to overcome in CAR NK cell therapy. Major roadblocks include their limited expansion ex vivo, lack of durable persistence and survival in vivo, inefficient trafficking and infiltration into tumour sites, and susceptibility to the immunosuppressive tumour microenvironment. While there are obstacles, potential strategies continue to evolve and may pave the way for more accessible, more effective, and safer CAR NK and CAR T therapies. Comprehensive solutions for CAR T and CAR NK therapy development are available, such as Sinobiological's one-stop reagents and CRO services, covering the entire process from CAR development to CAR T and CAR NK cell quality control. To find out more about CAR T and CAR NK cell therapies, check out our InFocus with Sinobiological on www.biotechniques.com.